Bismillahirrahmanirrahim. How are you, dear students? I hope that everyone be healthy. Today I am going to discuss the issue of 10th class, that is metabolism and enzyme. First of all, we are coming to define the metabolism. What is metabolism? All biochemical reactions that take place in a cell are called metabolism. What do we have here in the definition? Biochemical. Bio means life. These are the chemical reactions which are related to life. In living organism, in cells, they take place. So we can say all biochemical reactions that take place in cell. In cell, these biochemical reactions are called metabolism. Here we have some examples of the biochemical reactions, like photosynthesis. In photosynthesis, the food is made by autotroph living organisms. And respiration, we have digestion. When we eat food, the food is breakdown in our body. This is digestion. Growth, we are becoming growth and bigger. Our living organism are changing their body size. So that is growth. And reproduction. These are all the examples of metabolisms. And these metabolisms which I have give you the examples, they are occurring in the cells. We have anabolism in two categories. The first one is anabolism and the second is catabolism. First of all, we are coming to anabolism. That what is anabolism? Anabolism, monomers or small molecules of compounds, organic compounds. Here we have small molecules of compounds. Those are monomers. Monomers of starch we have here glucose. When glucoses are combined with each other, they make starch and water by the help of energy. That is a type of chemical energy. When chemical energy is used with glucoses and these glucoses or monomers are combining with each other, and after that the starch or a big compound or a polymer is made. This type of reaction we can say is anabolism. And where is it happening? In the cells of living organisms. Or simply we can say that anabolism is the reactions of building. When we build something, the meaning of anabolism is building of something, to make something. And catabolism is it has the meaning, opposite meaning of the anabolism. Catabolism. Catabolism means to destroy, to separate. How can we separate something here? Starch we have. Here we had glucoses and glucoses combining with each other, starch is made. But here we have starch. Now we are going to break down the starch to its monomers by the help of water. So catabolism, in catabolism, the big molecules are going to be separated or divided in its components. Those are glucoses. And in catabolism, we get ATP. In anabolism, we use ATP, we use it. But in catabolism, we get the energy. ATP is a type of chemical energy. So these are two types of reactions. When they happen at the same time in cells, we can say the metabolism is occurred in our body. Another issue, that is enzymes. What are enzymes? We are coming to define the enzyme. Enzymes are biological catalysts. If you remember from the class, the definition of catalysts, catalysts are those chemicals which accelerate the chemical reaction, but they do not take place in the reaction. So the example of catalyst is enzyme. Now here we have enzymes. Enzymes are biological catalysts. They are accelerating the chemical reactions in cells. Enzymes reduce activation energy. When a chemical reaction is occurring in the cell, in that time, if there is no enzyme in the cell, so the cell can use a lot of or too much energy. Now what do we have here? We have enzyme. When we have enzyme in cell, so enzyme reduce the activation of energy or activation energy and increase the rate of the reaction. In that case, if we have enzyme in the cell, at that case, this reaction is happening in short time. The time is decreasing by enzymes. And other thing we have, that is substrate. What are the substrate? The substrate are those substances that an enzyme acts upon. This is called substrate. This is the definition of the substrate. Substrates are those materials or those things in which the enzyme acts on it. So the substance that an enzyme acts upon is called substrate. Example, here we have maltose. Maltose is a type of what? Maltose is a type of substrate. Now for the maltose we have an enzyme. This enzyme is maltase. So maltase acts on maltose. So we can say maltase is enzyme and maltose is the substrate. Types of enzyme. How many types of enzyme do we have? We have two types of enzyme, simple and complex enzyme. What are the simple enzymes? Simple enzymes are composed of only just amino acid. These types of enzymes, we are dividing them according to their structures. Here we have two types of enzyme according to their structure, simple and complex. Simple enzymes are structurally made from just amino acids. The proteinic part we have in simple enzymes, just protein are there. But in complex enzyme, here is the example of the simple enzyme, that is pepsin. If we look to the complex enzyme, second type according to structure, complex enzymes are composed of amino acid, the first part. It is complex. First, amino acid. And the second component is non-amino acid part. It has two parts, two components, amino acidic part and non-amino acidic part. For example, catalase. Catalase is a type of complex enzyme. Now, this catalase enzyme is made from two parts, the proteinic part and the prosthetic part. The proteinic part, we can say it is the amino acid. Proteins are made up of amino acid, but the prosthetic group are other materials. What are the prosthetic group? Which type of materials are in prosthetic group? Here we can see here that prosthetic group, two types of materials are in prosthetic group. What are they? The first type of substance which are in prosthetic group, those are coenzymes. And the second part, the second type of enzyme, these are cofactor. What can be coenzyme? Coenzyme can be some vitamins like vitamin B. And cofactor can be minerals or ions. Minerals or ions. For example, calcium, potassium, magnesium. So these are the examples of cofactor. Calcium, magnesium and potassium, these are the examples of cofactor. And vitamin, like vitamin B, it is the example of coenzyme. Now, in complex enzyme, what do we have? We have two parts. The proteinic part, which are made up from amino acid, and prosthetic group can be one of them, not two of them. Just one of the component is coming in the prosthetic group. If we want to make a complex enzyme, there must be a proteinic part and the other part can be vitamin B. If we don't have coenzyme in this prosthetic group, in that case we can put the cofactor instead of prosthetic group. That can be a mineral. What are the minerals? Here we have some examples. Or it can be ions. What are ions? Ions are the elements with charges, or the small particles which have positive or negative charge. These are ions. So the prosthetic group can be any type of this ion or mineral. After that we can get a complex enzyme. Now we are coming how enzymes work. How enzymes work in our body. Enzymes can work like a lock and key model. If we look, it works like lock and key. Just look here. We have here a type of enzyme. Just imagine that it is the maltase enzyme, and here we have a substrate. The substrate is the maltose. Now, this enzyme must break down this maltose to its simple parts, which are glucoses. Here we have an enzyme, an active site. Which type of site? Active site. The substrate is coming and joining or combining with this active site. And after that, we can get this type of structure or model. Substrate and enzyme are combined with each other. This type of structure is named as enzyme-substrate complex. What do we have here? We have enzyme, substrate, and when they combine to each other, we can get a complex structure. So, this substrate now in the complex form. After that, the substrate is broke down. After the breaking down the substrate, the enzyme can be activating for the second time. There is no any change occurring in the enzyme. Now in that case, when maltose combine with the enzyme maltase, for maltose we have maltase. When they combine to each other, in this part, activation energy is reduced. Not too much energy is used. So enzyme can reduce the activation energy. So this is the example of how the enzymes works. Now look here. I have here key and lock. We talked here, they work like key and lock. Just think I have a lock and a key. Now this key is the substrate and the lock is the enzyme. This lock, now it is closed. I want to open it. For this lock, for this enzyme, here we have an active part. This enzyme is combining with the substrate. When it combines to each other, we can get enzyme substrate complex. Now we can open with this key the lock. Now it is open. So we can say this enzyme can act just only to this substrate, to this key. It cannot act to other key, to other enzyme. For example, here I have the enzyme, it is the substrate. Now this enzyme cannot act on this substrate. It is not going to inside, so it cannot work. So we can say enzymes and substrates work like key and lock model. Here we have some properties of enzyme. First one, enzymes decrease rate of activation energy. Here you can see that. When enzyme acts upon a substrate, in that case the activation energy is reducing. Second type, one type enzymes are one type of reaction. They just do their activation on just one type of reaction. For example, here we have maltose substrate, the enzyme maltase work on it. If here we have, for example, saccharose. For saccharose, there must be saccharase enzyme. Another key, there must be another key. So enzymes can act just on one type of reaction. And enzymes are used again and again. Again and again. The enzymes, when this enzyme maltase break down this maltose to glucoses, for the second type, the same enzyme can act on other maltose. It is continuing like that. So we can say that the enzymes used again and again. And enzymes work outside of the cell. Enzymes work outside of the cell, not inside of the cell. They are working outside of the cell to break down the materials or organic compounds to its monomers. After that, these monomers are entering into the cell. In that case, they are working outside of the cell. Here we have pH and temperature. These are two very important factors for the activation of enzyme, that how enzyme works. If we have a normal pH, power of hydrogen, basic or acidic solutions, if we have basic or acidic solution, in that case the enzymes can work perfectly. If this pH is changing, what is the normal pH? If you look here, the normal pH or neutral state of the pH is 7. When this pH is increasing from the 7, that we can say that materials are going to in basic state. In basic state. When the pH decrease from the 7 to 6, 5, if the pH of some materials become 5, we can say this materials has the acidic state. So it's belong to pH. So we have different types of enzymes which acts on the substrate. If the situation or state of the pH is not in the optimum state, in that case the enzymes cannot work. And also the temperature is the same. Temperature is also same like the pH. There are many types of enzymes we have, and each type of enzyme has its own optimum temperature. And another property that is very important, do not forget it, that is the term ASE, ACE. If we see in every term the ASE, in that case we know that it is the name of the enzyme. Like chitin. Chitin is a type of polysaccharide. For chitin we have a type of enzyme. This enzyme is named as chitinase. At the end of the chitin we are adding ASE. After that we can get the name of the enzyme. For example, here we have maltose. What? Maltose, substrate maltose. Maltase we have. Saccharose, saccharase, amylase. Many types of enzymes we have. So we can know the enzyme with ASE. And they are reversible. When they act to some substrate, after they are going to backside, they are reversing and they are reversible and they are active for the second time. And also we have another property that it's one substrate. They can act just on its one substrate like key and lock. At the end of the reaction, enzyme structure don't change. When this reaction is in the end, or it's go to the finish, when finish this reaction, after finishing the reaction, the enzyme cannot change. Now it is active for the second time. So it was your today's lesson. If you have any question, you can contact with me in WhatsApp. Have a good time.